Okay, so welcome back from our August break. I'm Janusz Hagush. I'm at Stony Brook, and I'll be sort of introducing the speakers today. So this is the N3C Community Forum, September 11th, 2023. Next slide. So just a couple of reminders. So welcome everybody back. Presentations will be recorded and will be archived in the Data to Health YouTube channel. So we have this recorded. And please use the Q&A button to post your questions. And then also use the raise hand feature for the host to unmute your microphone to ask a question. Okay, so key figures. This project, we've been going for a while, and we keep updating our data and adding new sites. Right now, we're at 80 sites. And we have 20.2 million individuals, almost 8 million COVID positive cases. If you look at the number of rows across the different domains, you can see them breaking down. We have almost 30 billion. So a very large, sizable data set that was built over the last several years. Next slide. So this is something that I was involved with, but other people I'm sure have been involved with too, so there is one of the additional features that have been added, and I think it's on our second sort of update recently. The vocabulary change is that we use the Odyssey vocabulary, and the Odyssey vocabulary is managed externally, and on a sort of semi-regular basis, they make changes to the vocabulary, and those sometimes can have effect on your analysis. So just a couple of, so you probably, if you author a concept set, you might have seen an email recently sort of detailing potential changes in it. So I'll just read it through. Concept sets analyses that relied on the older vocabulary need modification. Concept set authors will receive a notification indicating any vocabulary changes. I just got one recently, and in that case, the change was minor. Vocabulary change management is for concept set authors to see what changed and to address the changes. So in the case that I got, it was some titles had changed, so it did not affect my analysis. But if, let's say, a concept went from standard to non-standard, it might affect my analysis. And then we also have some great tools that are continually developed. The concept set browser and the vocabulary change explorer can help researchers find and acknowledge changes. So once you see a change, as sort of the concept set author, you should be going in, looking at that. If it's something minor, it might not affect the analysis, acknowledging it, or you might need to sort of make a change to your concept set. And you can see the link on the bottom if you want more details on it. Next slide. So we have upcoming forum presentations, and we've moved to a 2-week cadence for these. So the next one is going to be on September 25th, 2023, and Kate Bradwell is going to be updating us on the great work that she's been involved with the protocol pad. October 9th, it's a federal holiday, we'll have no presentations. And then October 23rd, Emily Fav will have a presentation on the black boxing health AI, demonstrating reproducible machine learning computable phenotypes using the N3C Recover, a long COVID model in the All of Us data repository. And this is going to probably be interesting because All of Us and N3C shares the Odyssey data model. So learn how sort of the idea of having a common data model, can that be leveraged for validation across different data sets? So that's going to be interesting talk. Next slide. Okay, so I'm going to go on mute soon, but today's we're going to have Jared Anzelin and Jeremy Harper provide some updates on the publication committee. So all the great research we do and sort of analyzing data, and we do that to extend our frontier of scientific knowledge. But there is a final step, as once you get your downloads approved and you write your publication, there's one final step in that process of going through the publication committee. So we're interested to hear sort of an update on the process for it.
My screen with the other
Slides really fast.
Right.
Hey, everybody can see it?
We can.
Mhm.
Thank you. So I'm Jeremy Harper, and I'm going to take the first half, and Jared's going to take the second half and close us out. Jared's put in a ton of work on all of this, and just really appreciate all of the effort that he puts in on and he's our newest publication committee member. So it's really been nice to have him. We'll be pitching that we have openings on the publication committee, and anyone who's interested in it, we'd love you to join us as well. Next slide. Oh, and I'm Jeremy Harper. I'm a biomedical informatician. A number of you know me. I've been working with N3C since it started. When it started, I was the Chief Research Information Officer at Reagan Street Institute. And these days I am a consultant who's popping around different projects. The publication committee, high level. Today we're going to talk through the publication committee process, the results, downloads, technical considerations, what you need to do to submit to the knowledge store, doing a few reminders, and Jared's going to talk a little bit about the chapter that has been published in the Researcher's Guide to N3C. Next slide. At the heart of the publication committee, we are here to facilitate your collaborations, to help you get out the door in a very successful manner. We are not here intending to block you from getting publications out the door. We are made up, the publication committee, of volunteers who are active participants within N3C. And so if you're ever feeling like the publication committee is a barrier, feel free to pick up the phone. We are happy to chat, talk through those concerns that we are finding. We have certainly streamlined the process. If you haven't gone through the publication committee a year for the last year, we are much better about here is specific documentation, specific actions that we are asking you to do. And one of the big things to remember is we are not peer review. Many of us are scientists at our nature, or have deep understanding of the data, and will often ask questions that peer reviewers might. But our first and foremost goal of going through the documentation is not peer review. It's to serve as a publication committee. That second bullet point of looking at we're looking at a wide variety of different things. We're asking you essentially, if you are going to publish about the N3C, it should be put in, whether it's a report, an analysis, an abstract, a presentation. Some things we do a very light review. So take, for example, presentations we ask you submit, but the full committee doesn't actually go look at the presentation or the presentation submission. Usually it is the person who's on call that week to review the new requests. Next slide. Again, in part, our charge, our mission, not peer review, but to ensure compliance with our requirements. Frequently, people do download requests that don't include any PHI, but once they get into the weeds, there are, and it will never include PHI, I should say, but the small cell sizes are often something that we have to be concerned about. So we're here to help facilitate getting the right attribution to the right groups within N3C. We're here to help troubleshoot authorship questions, and we also track the output of the N3C community. We're voluntary. We meet weekly. We have gone from an hour a week meeting that often went over to a half-hour a week meeting, with the occasional hour, not because you all are submitting fewer. We're just getting more and more efficient as we've gotten more experience with what type of things come up with publications. Next slide. Back one slide. So this is the publication committee. Swaroop and Carolyn are the co-chairs, and then the rest of us are just members. And so you can see we have 2 vacant seats. Please volunteer yourself, volunteer other people. If you know anyone, I have found this immensely rewarding. I get to see lots of brand new publications with new knowledge and tidbits about COVID-19. I've really streamlined my own process for what do I look for when I'm reviewing a paper. When I first started, I was taking 5-6 hours to review any individual paper. And now I've got it to something where it is much more manageable. It usually takes me half an hour to read through the paper and jot down coherent thoughts and processes. It's a great opportunity if you're someone who wants to know what's going on and see everything that is going on. And again, Jared's our newest member, and we're really glad to have him. Next slide. So I think most of you have probably seen the project life cycle overview, but just really quickly, there in the yellow on the left-hand side is you go assemble a team, you go get the DUR, the IRBs, you analyze the data in the enclave, you then go make a download request. That download request is not the publication committee, it is the data download committee who has oversight. And that is made up of NIH and leaders of N3C. You get your download bolus, it's provisioned to you, and then you draft your manuscript. You submit that draft manuscript and publication intent form over there in the blue, and that's where the publication committee comes into place. So we start looking and making sure that your DUR matches with what you're publishing. If it does not match, we make you go back and confirm that you actually have the authorization to publish on this material. We look for cell size constraints. There are a number of cell sizes, and we're going to go through that in slide 17. We make sure that the manuscript's actually about the National COVID Collaborative. There are times that people have put publications forward that have nothing to do with COVID-19, and this is the National COVID Collaborative, and it does need to have something to do with COVID-19 if it's not a pure informatics research style paper, where you're talking about the infrastructure. And then we also help collect the consortial contributions. We look for making sure that you're putting the right attributions in the paper, etc. Next slide. So there are some guiding principles that we've put down to keep ourselves accountable and the community to know what we're looking for. And so we're going to dive into each one of these over the next couple minutes. So I'm not going to bother reading them out loud because we'll get them into the next slides. So let's jump into the next slide. So our core responsibilities, data partners and individual patients. We want to ensure that when you've prepared the data for publication, it's consistent with the data use that you were approved for. So that's the DUR piece. Is what you're purporting matching with what you said you were going to go out and do. And we look for consistency with policies. So there are a number of things that we check for with that consistency. So the examples there on the screen, I'm just going to call out a couple of them. Cell sizes, obviously the second one, we're going to talk through in depth. It is one of our number one use concerns. The native population check. So if you're not aware, the NIH has specifically worked with the tribal councils, and there are specific behaviors and patterns and specific ways that we have leveraged the data So if you are calling out a native population within your research study, we make sure that it's in conformance with what has been agreed on that larger work. And we have experts. I'm not the expert in that to help guide you if you have questions. And we've got some of that mentioned within that book of N3C. We also make sure that it doesn't identify specific data partners. Frequently, we have people using all the internal ID numbers for data partners, and we make you go generate a 1, 2, 3, 4, 5 type list. And we're always looking for, is this actually focused on COVID-19? And there have been situations where we've had to go work with a publication to realign it so that it's looking at COVID-19 first, and maybe some of what they originally were considering the most novel and interesting bits of research are other observations that they have made along the way. Next slide. So we also look at your individual and consortial contributors' capabilities. This is making sure that there are a lot of people contributing to N3C, and that all of those different groups are attributed. So while we do our absolute best as a larger community to make sure that people are working within subdomains and teams, the publication committee is the group that finds out, oh, look, this one team was not working with anyone else, and they're maybe leveraging capabilities that other people have put time, effort, energy, and mental knowledge into. And so helping them give attribution where attribution is due is something that is important. We also make sure that if you're Recover funded, you're following and doing the Recover processes. This is often the piece that we really rely on Julie and Tersany to they take care of so much of this, of working back and forth and making sure the right people are attributed. Next slide. I am sure that most of you have seen this, but this is just in all the documentation describing what where you become a masthead author, where you're a consortial collaborator, and when you're in text acknowledgements. You will see that the people who are on the publication committee and who were the primary reviewers any given week will typically go in and fill out the information so that they become consortial collaborators rather than just in text acknowledgements, because they have dedicated more time that you might not always see. But like I said, it can take significant amounts of time to do a proper review of the papers that you submit. Next slide. We make sure that you've got your required acknowledgements and the data, the number of people who are coming to us without any acknowledgements has really slowed down because we've gotten a lot more into with giving you the right information as you're going to go submit to us. Julie did a lot of work last year to revamp all of the request forms. But even things like the IDEA program, if you are in one of these states, you need to acknowledge the IDEA program, and you may not be aware of it. And so those are the things that we are dotting all the I's, crossing all the T's. Next slide. We're looking at whether the N3C collaboratory descriptions are accurate. So if you haven't mentioned, if you are in level 1, level 2, level 3 data, we tend to say, hey, this is something that we need to know. If you are trying to represent that you have all the patient data from all 80 sites, we make sure we go correct you. And we make sure that you're looking at and acknowledging funding. Next slide. And then we do try and serve as a resource for authors. Now that we've got Jared, Jared or I will often jump on calls to help people troubleshoot through maybe more of the technical issues where maybe they think that they understand the data and there's some question of whether some of the assumptions are a safe assumption to make and what type of acknowledgements should be put in place if you're going to go forward with some of those assumptions. We do look for, okay, duplication, replication of other research studies, because we've read as a committee all of the different research that is out there. If the numbers are massively out of whack for what has been published in the past, we tend to catch it and we tend to call out and say, does this make sense with what has already been published on the N3C? Why doesn't it make sense? How do we make sure that these papers acknowledge, oh, look, this other work has been done. It reported this, we're reporting this. Here's the nuances and differences there. And then we work with authors for if authors have a concern, and we've had some situations where authors have had concerns about inappropriate use of their intellectual property, attributions of findings, data artifacts. We try and be that independent third party to keep everyone happy and honest. Next slide. Just your high-level submission overview. We want all manuscript drafts submitted. Like I said, a number of them don't wind up going to the full committee. If it's a full publication, it will go to the full committee. Podium presentations, posters, anything about N3C, we ask that you put it in. We've got a streamlined process so that it's not going to be a barrier in your way to getting the submission in. Just the authors and the committee piece. This is going over the same information that we've been providing, and just another way of you assimilating the data. We want to make sure that you're looking that you're in accordance with the DUR. We want to make sure you're looking at the download policy, the number of submissions that have gotten to us without a download permission. We do catch that, and then there are many conversations to be had because you have downloaded data that you agreed to never download. And that's one of the roles of the publication committee to make sure that your download matches what's going on with the data. In general, we don't go do deep dives into the downloads themselves unless there is a specific question over how did the download committee miss this thing that we've found that is potentially identifiable. But we do have a lot of access to be able to start going and asking those types of questions and raising those concerns to the governance side of the house. And let's go on to the next slide, because I need to give Jared his fair time. Remember, we do have policy. You do agree every time that you log in to be in compliance as you submit your brand new DURs in terms of the results download and the request process. And then the next slide, I think, is where we get into the nitty-gritty with some details of data, if memory serves. So if you can keep in mind, when you're aggregating data to small groups or small cell sizes, we don't want to be able to go back and recalculate what that less than 20 was. There are constraints related to native populations. Zip codes and regions representing rural populations of 20,000 or less are fully obscured. Zip codes and regions of more than 20,000 persons that overlap with tribal communities are identified as AI/AN, are identified by the first 3 digits. And then remember, there are constraints related to data partner IDs. And then the next slide. And this is a specific thing that we now hand out to help put specific information in front of you so that you know exactly where we might have been concerned with some of the ways that you handled small cell sizes. So if we go to race and ethnicity and AIAN, you see in the very first original, we've got 14, a 6, an 8, and they're marked in red, yellow, and red. Those are less than 20, so that is a concern for us within the N3C. Insufficient masking. This is something that we see frequently. You can see that in the two highlighted yellow cells, those, if we look at the columns, they are only a single instance of less than 20 in that entire column on either side. So that means that if I add up others and white non-Hispanic and white Hispanic and black non-Hispanic and black Hispanic, I can go to that total number right at the top of the page where it says 5,000, subtract all of those other totals from 5,000, and I will have what that less than 20 wound up being. In comparison, I could add up with the green cell of COVID plus, that column, and I've got two less than 20s. I might find out that there are 18 people who are in the less than 20 group, but I'm not going to know if that's one black Hispanic or 17 black Hispanic or 18. So we will have you go and mask multiple cells. Most investigators, if we go all the way over to the right for sufficient masking, will choose to do something like here, where rather than 62 and 72, is that 60? I've got to make this 82 and 72, less than 85 and less than 75. We've seen investigators skew data. We've seen investigators do a lot of different things to handle the data, and we won't dictate what you do to handle the data. What we will do is make sure that you are handling the data in such a way that it does respect the less than 20. A new policy that has just recently come out is, in the past, take gender, female, male, missing or unknown. If you had missing or unknown, you would have had to obscure another cell or done something. And there are a number of, usually it wound up being you were going to skew your other numbers. Recently, the governance committee has authorized missing and unknown to we are going to mask it with the less than 20, but we are not going to require you to do other things for that missing and unknown category. To some extent, that is it's already missing and unknown. So with that, thank you all for your attention. I'm going to hand it over to Jared.
Great, thank you, Jeremy. And I see we did have a question in chat. Not sure if Ken Gersing is available. It's more of a question for him. Question is, we only have 6 data partners, viral variant information, and it is mentioned that 9 other data partners will be sharing that soon. Do you have any updates on that you'd like to mention to the group?
Yeah. So it's a really good question. When COVID was really going gangbusters or whatever, that's not a great expression, but it was really peaking and we were getting a lot of changes in variants, the community asked us to get viral variant data, and we took some, we were able to find not a lot of money, but $20K to give to 9 sites. That's all we had. And 3 of them either just couldn't do it or couldn't do it. It was pretty complicated in that we had to, often the system that had it was actually acting on the behalf of the state. So we would get that data, and then we, and we got 2 pieces of data. It was kind of summary data, and then there was actually the sequence data. So the sequence data was placed into the National Library of Medicine. because we don't want to reproduce the work that they've done with holding genetic data, and it was linked to N3C. But the call for that has been pretty minor. And so that data is available. And there's actually 4 other potential sites that we were given from, I'm not going to remember the institution's names. I'll get it wrong. I think it was Mayo, University of Texas Methodist. Why am I blocking on the other 2? They'll come to me in a second, but that we did get. So there's actually 4 other sets that are embedded in N3C. Nobody's ever asked for them, that are available. We don't have any more funds to go get other viral variants. So that's the long and the short of it.
Great, thank you. And then we had one more question to confirm for gender. If it is unknown or less than 20, we can report that as less than 20, even though you can do the math. Yes, that is correct. Does not apply to other sex or gender categories. That applies to strictly missing unknown data as an FYI. And then Jeremy had touched on this before, but in terms of obfuscating data partner IDs, N3C has persistent data partner IDs for sites that contribute data to N3C. And while these cannot be used to directly identify sites, they do persist across N3C releases. And so we require them to be obfuscated before being presented in any tables or supplemental tables in publications. It also behooves you to say that you really shouldn't be singling out sites in your publications. So not all site level analyses are appropriate, but there are times when they are. It is never appropriate to attempt to re-identify an N3C site or tie a site to a specific geolocation. And so when you present site-specific findings, you should present them with a site or a study-specific identifier. So if the study site had a data partner ID of 1, 2, 3, 4 as an example, that would become site 1 in your publication-ready preparation. Next slide. And we have another question. Can I remove missing other gender from analysis? That is a scientific question. If it's important to your statistical analysis, I guess we would recommend dropping missing/unknown data if you're using it in modeling, but otherwise that's a scientific question. In terms of technical considerations, and a lot of this is outlined in the chapter in the guide to N3C, so I'm not going to go into it in great detail, but there's some very technical considerations when putting together data to be downloaded and presented in manuscripts. One of them is pinning to a release. Since these analyses take often months, some longer, unfortunately, to complete, you want to make sure that all of your data download requests are from a specific N3C release. One of the amazing things about N3C is the scale, and data comes in constantly and it is released on a weekly basis. So if you are constantly pulling data from the master branches, it's called, that data is changing and evolving over time. So making sure that you pin your analyses to the same release becomes very important. We also recommend in supplemental materials or somewhere in your paper indicating which release your results come from, just for replicability. That makes it a little bit more scientifically transparent. Next slide. And Jeremy had alluded to the download review process. That is not a job of the publication committee, but it is part of the publication review process. When we receive manuscripts, we make sure that you've checked all the boxes and any results presented have gone through the download review process. We also secondarily review those to make sure that no small cell counts can be back calculated and no small cell counts are presented in either the body of the manuscript or in any of the results. So that is part of the publication review process. Next slide.
Jared, I just wanted to add something to something that you just said. it's very unusual that, so every week on Monday, I'm really just reiterating or what Jared said. Every week on Monday, all data sets are redone from beginning to end. So it's over 27 billion rows of data and versions. So I just want to reiterate, because if people don't understand that they're not appended, they're start to finish redone. And each one of those releases has a very unique identifier. And it's totally fine that let's say we've got 40 releases out or 140, doesn't matter. And you've been doing your analysis on release 139 and 140 came out today or next week. It's fine to stay on the one you're on, but you also can switch to the newest, the latest, and the greatest, which hopefully has new data in it. But I just want to make sure the folks understand that each release, it stands alone as it, and each one of them has release notes on it. So if there's something special, I don't know, this site was bad and we don't, whatever, you would see something like that. So I just want to make sure that people understand they're not appended, they're really unique instances. And the reason we did that is because we want you don't have to bother, combining sets and, oh my God, what did I miss? And why do I have two row 10s? That's all taken care of but you're moving your analysis and pointing it to another set of data. So I'm sorry, I just want to make sure that people understood that.
No, and that was great. And we do have in the guide chapter, we have specifics on resources for how you can pin a specific code workbook or results to a release version, just as best practices, and that'll save you some frustration as you work through projects. Next slide, thank you. And in terms of code sharing, there is the ability to download a code workbook, the code in it, to a GitHub repository. We have some documentation on how to do that. While the publication committee does not directly review those repositories, if they are presented in the manuscript, we will do our best to at least go look through them. The same rules apply. No presentation of person IDs or data partner IDs are allowed in a GitHub repository. So if you're extracting code and manually singling out sites for inclusion or exclusion, you would need to obfuscate those if you're presenting any code that comes out of N3C. Next slide. And concept sets are a unique download class within N3C. The concept set browser allows you to export a list of OMOP concept IDs that are associated with a concept set and take those and run, so to speak. Those are all public information. They don't have any patient-specific information or counts. But if you want to present those along with your findings for transparency, that is something you can do. And there's an export feature that's tied to a specific concept set and a specific version within the concept set browser. Next. And then the same is true with the knowledge store. If you want to share some reusable code with the N3C community or present it in a way that others can replicate your work, the knowledge store is a great way to do that. And that gives you the opportunity to share your code, share computable phenotypes with the community. And there's a full process to publish objects in the knowledge store that can then be imported into other projects. There is some attribution that goes into that. So if somebody pulls in a knowledge store object that has been created by the community, Jeremy had alluded to the consortial collaborator process. There is a process for those people contributing intellectual property to kind of be acknowledged in papers at the consortial level if they've done a substantial amount of work developing code that's used by others. Next slide. And then the publication intent form. This is a newer process that Kate Bradwell has spearheaded over the last year, and it is officially live. So now all of the publication intent forms are submitted within the enclave, and this does a really great job of taking a lot of the technical work out of your hands and putting it into the platform. You're able to link directly with manuscript metadata within this form. So any of those download review requests, once approved, they can be directly identified within this this publication intent form. Your data use request and the level of access are all acknowledged within there, and then you can acknowledge any concept sets used or knowledge store objects used, so that all of that metadata can be gathered in one spot. And a few months ago, there was an N3C community forum on this, so I'm not going to go into it in detail, but it's covered there, and it's also covered in the guide chapter. Next slide. And then I went through that quickly, but this is one of many chapters in the Researcher's Guide to N3C. I highlighted our chapter here, but there are a significant number of really in-depth chapters within the Researcher's Guide that cover everything from team formation and team science at scale in N3C through curating concept sets, actually analyzing the data, and then going through all of the steps to move towards publication. So I would highly recommend checking that out. And next slide. This is the group of authors. I think it might be a little out of date that have done a lot of the contributions to the guide to N3C. Those underlined are on the editorial committee, which is led by Sean O'Neill and Will Beasley. And so kudos to them for making this happen. It's really a truly tremendous resource. It's modeled after the guide to Odyssey, but specific to N3C and the unique considerations within N3C. Next slide. Thought I had another one here, but these are the important links. I thought I put a collaboration map on here, but I did want to also thank the community. It's kind of a staggering amount of work that's come out of N3C. Hundreds of publications from hundreds of institutions representing thousands of scientists. So that is, as much of a thank you to the community for all the productivity and collaboration over the last 3.5 years. And then big thank you to Tersunet and Julie McMurray for making this publication committee function.
I also want to thank everybody. I am echoing what Jared is saying, and Jeremy, an unbelievable amount of help from everybody. Thank you so much.
Yeah, any other questions?
We did have a question come in that may not have an answer from anyone here, asking about long COVID. Apparently the pre-populated data table had about 200,000 people in it. And after the latest release this week, it got reduced to 180,000 people. And they were asking if there was a specific reason why there was a 20,000 person loss in long COVID.
Jared, we are aware of that and looking into it. We know there's something is amiss, but we're not sure. We as of this morning, we know for sure that it wasn't, unless we missed it, it wasn't a Palantir thing where people maybe were dropped because the site hadn't given data for a certain number of times. So it seems to be from sites, from somehow the phenotype has changed and the data that's been moved over from the sites, which doesn't really make sense, because if you're, obviously this started pre-COVID in January 9th, 2018. So we are aware of it and we are looking into it, but I don't have an answer.
Thank you. That answers their question.
Yeah, and for researchers, like if you do see anomalies, right, you can submit a bug report on it, right? So it's an issue. So Are there any other, I don't see any other questions, right?
We have a hand raised. I don't know that I have the capacity to unmute someone, so maybe Lindsay, if you can, there we go.
Hi everyone, my name's Lindsay and I'm a student, so I'm very new to the N3C and I'm not sure if this is the right place to ask my question, but I'm getting ready to submit an abstract for a conference later this year, and I just wanted to kind of clarify the download policy and then how that correlates with me like sending my abstract in for review. So all of my data is just like percentages of the population. And it's all basically just like number over population, like just basic percentages. So I didn't know if that needed to go through like a download request or how exactly I would kind of go about that. And then what the like turnaround time is for like an abstract request to like be presented.
Lindsay, that I'm one of the people who does the download requests. And I try to look at them every day and try to get them out as soon as possible. So usually, but it's reviewed by 2 different groups, and we did that because we wanted have second set of eyes on, and we still miss things, and then the publication finds them. So, but yes, you do have to download it. We will try to get to within 24 to 48 hours. And the reason you're doing it, and we do appreciate the percentage, and we understand, but our promise to the sites that are giving us this data that we would assure, that there would be a set of eyes on this, because, all the sites, the data contributors really have bent over backwards to help, to allow us to have this data. And so this is kind of one of our promises. So you do have to do that. So please do it. And it is right on the homepage, there's a button that says, I can't remember what the label says, but download request, and it'll walk you through it. Is that?
And can I download like an entire like table that I have combined multiple tables for? Like, is that appropriate?
I leave that, Jared. Jared has more downloads than any person on earth right now.
Yeah, there are a few tutorials on this, and happy if you reach out to me. I don't know that I can post my email in chat here, but happy to walk through that process. There are a few different ways that you can compile like request to submit. They have something called Fusion that's kind of like Google Sheets within the platform, where you can just copy from your results within the platform, still within the platform. Or you can directly modify and censor small cell counts within a code workbook or within Contour, and then submit those for download review. And then the publication committee is very responsive to abstract requests for conferences. Our review time is 2 to 3 days on those. Can't make promises, if there's a holiday or something, but we require 2 reviewers for abstracts at the conference level, and then for full publications, they go before the entire committee and are reviewed by the whole committee. So those take about a week.
Okay, perfect.
It's very quick, yeah. And if we have requirements, we'll get you feedback. The committee meets on Thursdays. Usually Tersenate will send out feedback on Friday or Monday if there are requested changes.
Okay, perfect. Thank you all so much. I really appreciate it.
Yep.
I just posted Jared's email address in the chat, so everybody in this session should be able to see it.
I don't think we have any other questions, but thank you for everybody for coming back. A lot of this work is, behind the scenes detail work that people have been working on to make this amazing data set available for research and insight. So thank everybody that's been involved with that process. And thanks for the update, and the work you guys have done, streamlining the process. Okay, so we'll see everybody in 2 weeks. Thank you.
Thank you.
Thank you. Bye.